បច្ចុប្បន្ននេះយើងកំពុងតែជួបនឹងវិបត្តិមួយគឺទី1គឺវត្តមាននៃឱសថខ្លាំងៗទី2គឺឱសថដែលនាំចូលដោយមិនស្របច្បាប់ឱសថដែលមិនបានចុះបញ្ជីការហើយនឹងការចែកចាយឱសថដែលមិនមាននៅគុណភាពនៅកន្លែងចែកចាយឱសថNo sticker made in Vietnam.
Everywhere in this market, we know that they sell counterfeit and unregistered medicine. When we collect samples for testing, sometimes we purchase as we are normal consumer. But at the time, we come as we are official drug inspector. And when they see us approaching, they often hide the medicine in the drawer.
An ever-increasing quantity of counterfeit and substandard medicines is flooding pharmaceutical markets worldwide. Especially vulnerable are developing countries, which often lack the regulatory and enforcement resources to adequately monitor the production and distribution of local and imported drug products. Customs agents and drug inspectors are restricted in their capacity to police the pharmaceutical sector due to human resource and financial limitations. According to recent official World Health Organization figures, drug counterfeiting represented approximately $60 billion in sales. This represents almost 10% of the world pharmaceutical market. Unlicensed and unmonitored drug sellers, such as these seen here in Cambodia, are rife with substandard and counterfeit medicines. Access to quality health care is a fundamental human right. Yet often, patients in the poorest areas of the developing world are subjected to the dangers of counterfeit medicines, which can have devastating health effects. Essential medicines, such as antibiotics and antimalarials, are frequently and cheaply counterfeited. Recognizing the need for field monitoring of medicine quality in resource-limited countries such as Cambodia, the United States Pharmacopeial Drug Quality and Information Program established a network of sentinel sites in Asia, Africa, and Latin America. With financial support from the United States Agency for International Development, USPDQI provides mini-lab test kits produced by the Global Pharma Health Fund. The mini-labs are easy-to-use, portable, and cost-effective screening tools that identify medicines with quality defects. They provide all necessary lab components to analyze drug products collected in the field by local drug inspectors during post-market surveillance. USPDQI and local field staff conduct field surveys at both public and private pharmacies and at unlicensed drug seller outlets to sample and test antibiotics, antimalarials, anti-inflammatories, and other essential medicines.
So just as we are set to leave to do a supervisory field visit in the provinces, we received a report from the Pharmacovigilance Center that there's a doctor in Battambang who is having some problems with a patient. Apparently they're suspecting that the medicine that the patient took might be counterfeit or substandard. So we've changed our plans and we're now going to leave the capital and go into the province and see if we can find out if the doctor can provide any information about where the product came from, so we can follow up with this and find out exactly what the situation is.
It's the next morning in the countryside. We're traveling west into the rural provinces towards the Thai border, which is a hotspot for drug-resistant malaria in Southeast Asia. Poverty and counterfeit medicines form a double threat here, where more than 50,000 cases of malaria were reported last year. Patients must rely on unlicensed drug sellers for their medical treatments, often purchasing counterfeit medicines which proliferate in these impoverished rural locales. As a result, patients succumb to curable diseases, and the prevalence of substandard and counterfeit medicines can contribute to the emergence of drug resistance in the deadly malaria parasite. The criminal traffickers who produce and distribute fake medicines hold no regard for the victims of their crimes.
Okay, so we're just arriving at the Tasan Health Center in Samlot District. This is a public health inpatient facility, which is actually quite rare in these rural areas of Cambodia. The number of health centers depends on the population density. In this case, in Cambodia, the number of clinics is 1 per 10,000 people. Morning. Good, sure. Morning. All right.
បាទជាមួយនឹងក្រុមការងារមកពីខេត្តបាត់ដំបងហ្នឹងពួកខ្ញុំមកតាមដានការព័ត៌មានមួយដែលអត់លោកពូថាស្ថានភាពជំងឺហ្នឹងមិនដែរលោកពូអាចអនុញ្ញាតឲ្យមើលជំងឺបានទេអូចាអូម្នាក់ហ្នឹងឯងឈើ
lâu con mà đòi dương chẳng hay không tăng tai hà còn prai nó cứ là sọt mình có cả để tập lại nữa
cho chẳng chẳng lục bô miên nó khơi nam kia miên phải hai nào để cái phở ấy
miền
tuột
vô mà này
xong mơ thì chứ
nam bọc hết này bà
nam cuốn chanh thời bọc kê ta ông nam cuốn chanh nhá nè vẫn nó sao là mà tay nào tay you see it's anti-malaria
Oh yeah. So can we take a sample of this to the village to?
Phụ nhóm ai sống tháng năm nâng chứ sao là mà tay nào tay
bà sao là một tay nâng
chứ phụ nhóm vô thư tay ọt khỏi này ông dư thời tinh nó xa xa mình là chết nâng á
miền ta là miền
chứ dư ai hổ tinh ba này chứ
bà
ta nó nhóm xong tục ba đấy vừa thư cùng lưu
bà
ông cơn trời
bà
Okay.
cái kho là phụ nhóm vô nha
cái
tôm này tiên mít miệt lý à cha cha miếng cái kho là tiên đó
It may be difficult to find the same batch of this medicine from the drug seller in town, but we need to identify where it was sold and test it to ensure that other patients aren't exposed to it.
We're informed by the doctor at the health center that the patient failed to respond to treatment and has gone into a coma over the past 24 hours. He's not exactly sure where the medicine was purchased from, but he said that it was from the local village here. So we're going to head over there and sample from all of the drug sellers and try to determine if perhaps the medicine was counterfeit or substandard. It's in villages across the Mekong, such as this one in rural Cambodia, where unlicensed drug sellers outnumber licensed pharmacies. Impoverished communities lack the necessary resources to provide adequate medical care to patients. So oftentimes patients will go to unlicensed sellers such as these for diagnosis and access to medicines without ever seeing a doctor. So patients that are unable to afford expensive medicines are at a higher risk of exposure to counterfeit or substandard medicines which proliferate in rural areas. So let's go and see what we've come up with here. Okay, so this sample looks to be identical to the one that the patient purchased from the health center, correct?
Yes.
Okay.
Bông miên lụa chở nè tay năm nữa em cho qua buôn phèm bát té ha hay tuần lại mà đấy tuần lại buôn quan pram xôm vô bay bát té thiệt máu.
Yeah, so let's go ahead and collect enough samples to be able to test them back at the lab. Okay, great. Why don't you get the data collection sheet out here? So what we have here are samples that are identical to the one that the patient at the health center had purchased. So in this case, what we'll do is collect these for testing back at the lab, and we'll also do a basic visual inspection test here in the field and fill out all of the data collection forms necessary for reporting back at the lab. We're here at the Provincial Health Department of Battambang, and I'm joined by Mr. Asani, who is an officer of the Drug Bureau, Mr. Rit from the National Health Products Quality Control Center in Phnom Penh, and also Ms. Boravan from the Essential Drugs Bureau in Phnom Penh. This is one of several sites throughout Cambodia that are equipped with the GPHF mini lab. In addition to Cambodia, Sentinel sites are located throughout the Mekong region of Southeast Asia, Latin America, and Africa, supported by USPDQI and USAID for medicine quality monitoring. These kits are equipped with all of the necessary materials to perform the 3 basic field screening tests according to USPDQI protocol, including visual and physical inspection, simple disintegration, and thin layer chromatography, also known as TLC. Now we're going to test the samples that we collected from the unlicensed drug seller in the field.
In visual inspection, we closely examine the product container and the labels on the packaging and blister packs. In compliance with good manufacturing practices, also known as GMP, the manufacturer is required to include items such as the drug's trade name, the active pharmaceutical ingredient, known as API, manufacturer's name and address, drug strength, dosage form, lot number, date of expiration, and other important data. In physical inspection, we examine the characteristics of the tablet or capsule in solid dosage forms. Important characteristics to consider are uniformity of size, shape, and color, coating type, markings, scoring, or letters imprinted on the capsule or tablet, and any abnormalities such as cracks, breaks, color spots, or inclusion of foreign matter. In this example, the product on the right is genuine with a hologram on the blister foil, whereas the product on the left is counterfeit and lacks the appropriate hologram, thus failing visual inspection and would be sent for further analysis at the national lab.
At this point, we've determined that the product has passed the visual and physical inspection tests. Even though it's passed, it's not enough information to determine if the medicine is of good or bad quality. Now we'll continue on to the next test, which is simple disintegration.
Simple disintegration is meant to ensure that a medicine can release its contents into the body for proper absorption. Passing disintegration is one of the key components used to determine if a product is of good quality. In 100 ml of water, all coated and uncoated tablets and capsules must disintegrate completely within 30 minutes at 37 degrees centigrade, which is roughly body temperature. The batch passes the test if all 6 tablets or capsules disintegrate within 30 minutes. During this time period, the bottle containing the tablet or capsule is gently shaken until the product disintegrates. If a drug product does not pass this test, there is a major defect in its quality of manufacture.
The simple disintegration test results conclude that the sample from the unlicensed drug seller passed, and all 6 tablets disintegrated within 30 minutes. For illustration, we wanted to show a sample of a known counterfeit artesunate that fails to disintegrate within 30 minutes. As you can see here, the water is still clear and the tablet has failed to disintegrate. Next, according to the protocol, we'll perform thin layer chromatography tests on the samples from the unlicensed drug seller.
Thin layer chromatography, known as TLC, is a simple technique used to separate components of a medicine. It is a useful tool for identifying if the product contains the specified drug as claimed on the label. By closely following the TLC monograph for a specific medicine included with the mini-lab, lab personnel can conduct the test as simply as following a basic recipe. The active pharmaceutical ingredient in the field sample is compared to a known chemical product called a reference standard. Reference standards are highly characterized specimens of drug substances for use in conducting official tests and assays. The sample and standard are analyzed after development with reagents on a TLC plate. First, the analyst prepares the developing chamber for the TLC plate. This jar contains the mobile phase solvent and a filter paper wick, which aids in ensuring vapor saturation in the atmosphere of the tank. The chamber must be allowed to come to equilibrium over a period of 30 minutes. In the next step, all solids, including the drug sample and reference standard, are dissolved into a known quantity of extraction solvent such as methanol in preparation for spotting onto the TLC plate. The plate is marked at 1.5 centimeters from the bottom to ensure that the spots are uniformly spaced and that the spots will not be submerged into the developing solution in the next steps. Next, we see the analyst spotting the TLC plate with the drug sample solution and the reference standard solution. To ensure the spots are dry and uniformly spaced, they are checked using an ultraviolet lamp at 254 nanometers. The plate is then carefully placed in the TLC developing chamber, and the solvent is allowed to wick up through the plate. When the solvent front has migrated 3/4 of the way up the plate, it will be removed from the chamber and the solvent front marked. In the case of artesunate, as we see here, the plate will be stained with methanolic sulfuric acid solution in a petri dish, and then the plate is heated on a hot plate until the spots are visually evident. Finally, we analyze the spots on the developed plate, calculating the sample error percent using the retention factor, or RF, of the sample and reference standard. If the samples fail, the test must be repeated. After performing the thin layer chromatography test on our sample from the unlicensed drug seller twice, we determined that the sample has failed TLC. This is evidenced by the fact that in lane 2, you can see there's no spot and therefore no active pharmaceutical ingredient in this product. Because of the fact that it failed, it will now be sent to the national lab for confirmatory testing. The National Health Products Quality Control Center in Phnom Penh is where confirmatory testing takes place, and we've brought the samples here for analysis using validated methods, which are described in pharmacopeial monographs such as the U.S. Pharmacopeia National Formulary, the International Pharmacopeia, and others. After analysis of the sample according to the monograph, we compare the peaks of the standard and the sample as seen here. Based on the test results that were performed here at the National Lab, we can conclude that this sample definitely failed. A formal report of the results of this test will now be sent to the regulatory authorities, who should then take appropriate legal enforcement actions and product recalls. The Interministerial Committee on Counterfeit Medicines issued a national recall on these failed products after reviewing the formal report from the National Lab. With drug inspectors and the police, we've now returned to the unlicensed drug seller to enforce this action by issuing a formal warning and confiscating the product. Information about the dangers of this counterfeit medicine will be distributed to pharmacies throughout the country. The malaria patient from the clinic never regained consciousness and passed away. If he simply had access to good quality medicines, could have been easily avoided. Where do these counterfeit drugs come from? Who is manufacturing them? How do they get mixed into the pharmaceutical supply and distribution chains? With the proper tools, perhaps we can begin answering these questions.